Now I'm super happy to announce Lou Hawthorne, who is the founder and CEO of Nanotics, which is a novel nanomedicine platform creating nanoparticles to treat cancer, autoimmune disorders, and other diseases. Welcome, Lou. Ah, there you are.
Hello, everybody. And I want to thank Aubrey and Martin for inviting me back. I presented last year, and a number of you are looking forward to updates. We have updates both in terms of our preclinical collaboration with Mayo Clinic and also updates on our scientific thinking. And one of the things I love about Aubrey's conferences is that it's not just about data. You're also encouraged to share, kind of open the kimono, I don't know if you could still say that, and share what you're thinking about these days. So I'm the founder of Nanotics and also the inventor of our core technology, which is called Nanots. This is a new class of medicine. It treats disease and potentially aging via depletion of soluble factors. And nanots invert the drug paradigm of the last 2,000 years. Because rather than adding bioactive molecules to the body the way drugs do, nanots deplete bad signal molecules from blood that drive or enable disease. Nanots are really quite simple, they're fast, and they actually work. So the way nanots work is that there's a core, which is a structural element upon which we build functionality. It's completely biodegradable and scalable. Onto that, we attach capture agents that capture the soluble targets from blood. These capture agents never circulate the way a drug does. They're covalently bound to the core. Onto that, we attach a porous shield, and this is central to both the safety and efficacy of nanots. The shield enables soluble targets to diffuse through and get captured, but it prevents any interaction at all with cell surfaces, including proteins that are biochemically identical to the soluble form, and that's something drugs can't do. And finally, we attach a stealth coating to delay immune recognition and target clearance until the capture process is complete. It ends up being 150 nanometers across. You inject it like a drug, but it's cleared naturally by macrophages. It's basically a molecular sponge, a programmable sponge, or a molecular apheresis is another way of thinking of it. And we build them using click chemistry, which won the Nobel Prize last year. That's modular chemistry, so you can iterate really quickly and easily to new nanots against new targets, potentially for new diseases. Okay, they're very fast. They can deplete whatever they're programmed to deplete biochemically by more than 98% in less than 5 minutes. Compare that with pheresis, which is the closest analog. That takes 3 to 4 hours to deplete targets from blood by less than 70%. The safety profile is amazing. We haven't seen any mortality or morbidity in rodent studies, even at 100 times the planned dosing. So what can you do with nanots? All right, so the first thing is to get away from the cell-centric view of disease. We've been able to see cells for 300 years, and most of our drug strategies involve some intracellular pathway. That's not our domain. We don't target cells, we target signals. Cells do what they're told when they receive signals from other cells in general. So the diagram I'm going to show you used to be our most secret confidential diagram. We call it killers and chillers, and it's a map of our domain in oncology. The red rectangles around the perimeter are cells. They're killer cells. They produce killer products. Type 1 macrophages, NK cells, CD8 positive T cells. The blue rectangles are chillers. They produce immune inhibitory or suppressive products. Cancer cells do this, type 2 macrophages, myeloid-derived suppressor cells, and Treg cells. None of that is our domain. Our domain is in the center. Those circles are the soluble products produced by the cells. The red circles are inflammatory cytotoxic products, and the blue circles are soluble immune inhibiting products. And the page icons are third-party peer-reviewed publications that document that facet of this hypothesis. We're going to make nanots against all of those circular targets for treating a variety of diseases. Okay, so I mentioned the Mayo collaboration, and this involves a soluble target on the PD-1, PD-L1 axis. So membrane PD-L1 is a well-characterized molecule that's inhibitory on the surface of cancer cells. It's the focus of 3 approved drugs generating $6 billion in revenue. PD-L1 is an inhibitory protein. It ligates its cognate on the surface of multiple immune cells, which is called PD-1. And this receptor is the target of 4 approved drugs generating $29 billion. And the 1,000-pound bear is pembrolizumab, also known as Keytruda, which is going to be the best-selling drug of all time this year, $23 billion a year. Okay, but there's a soluble form of the inhibitory protein. Nobody's targeting that because they don't have a technology for doing so. And that's called SPDL1, or soluble PD-L1. There's some very good reasons, both in terms of safety and efficacy, to target this inhibitory species. Okay, so we're targeting this in cancer, and I already covered those first 3 points, but soluble PD-L1 correlates very negatively with survival. Many normal cells and immune cells, not just cancer, express membrane PD-L1. So the cells that express it normally as a don't kill me signal, the drugs interfere with that, and that's where the very serious toxicity of the PD-1 and PD-L1 drugs arise. Immune cells also use PD-L1 as part of their don't kill me signaling to prevent fratricide from other immune cells. So the PD-1 and PD-L1 drugs can actually reduce immune competence. So we're working with Mayo Clinic on Nanots against soluble PD-L1 for treating metastatic cancer. It's a great collaboration. The team actually includes the co-discoverer of PD-L1. And a world first announcement, which is that Nanots against soluble PD-L1 induced profound tumor suppression in humanized mice with triple-negative breast cancer, which is very hard to treat any other way. And Mayo reported, quote, "Our work demonstrates that selective capture of tumor-derived soluble PD-L1 with a novel nanotherapeutic platform," that's us, "can elicit local and systemic anti-tumor immunity." And that's being presented in November at the Society for Immunotherapy and Cancer in San Diego. I'm authorized to say what I just said and show this next thing, and that's it. So this is the classic graph with the hockey stick of tumor growth in the saline control, and then nanot inducing total tumor suppression. So it shows that soluble target depletion can be quite powerful. This is a monotherapy. There's no drug involved. All we're doing is taking something out of blood. And it also shows specifically that SPDL1 depletion by itself is efficacious. What's really interesting is that the drugs against PD-1 and PD-L1 don't work in triple-negative breast cancer, either in the animal models or in people, which raises a really interesting question. Why do nanots work better? Because you would expect that an antibody against membrane PD-L1 would also neutralize soluble PD-L1. So what's going on here? Why do nanots work when the drugs don't? There's several reasons. I'm going to touch upon really the main one, which is that nanots clear targets without stabilizing or amplifying. And this is the dirty little secret around antibodies, which is that they can actually generate an escalating reservoir of the very target they're designed to neutralize. And here's how it works. So the blue dots are tumor-generated immune inhibitors. There's a membrane form on the cell. There's a soluble form in circulation. When you put the antibody drug in, it engages in dynamic equilibrium with the target. So the target is bouncing on and off the antibody. And when it's on the antibody, it's neutralized. But for some period of time, it's off the antibody. And at that point, it's bioactive. Now, you're dealing with a target that normally clears in a few hours, and you're dealing with an antibody that has a very long circulation time, a half-life of, say, a month. So what ends up happening is the cell keeps secreting more and more of this target, the antibody keeps collecting it, and the dynamic equilibrium generates this larger and larger reservoir. The aggregate of target that's off the antibody at any one point grows. And that doesn't happen with nanots because nanots do irreversible target clearance. The nanots collect the target, and then they themselves are cleared by macrophage phagocytosis, at which point the target is gone. There's no reservoir generation. Okay, so how serious a problem is this? Well, let's look at atezolizumab. So the green line is atezolizumab concentration in blood when dosed every 3 weeks, and the red line is the total soluble PD-L1, both bound to the antibody and free. It's been documented in human patients that total SPDL1 builds up to 30 times the level that it was at the original time of dosing. What that means is that in trying to neutralize a membrane immune inhibitor, they are generating a massive quantity of a soluble immune inhibitor, which in general they're not even tracking. This leads to large spikes in the soluble free PD-L1. And that's important because there's a threshold of concentration that determines mortality. Above this concentration, which is about 800 picograms per ml, the survival of those patients is much lower. Contrast this with how nanots work. It's very easy for nanots at a regular dosing interval to keep the soluble PD-L1 concentration below that threshold of mortality risk because it's irreversible target depletion. Okay, so I want to talk about my absolute favorite biological axis, which is the TNF axis. So TNF is the master inflammatory cytokine in the body. It is involved in, we think, pretty much every non-genetic disease, whether it's on the inhibitory side, cancer, or on the inflammatory side. There are actually 6 faces of TNF, and in order to develop an intervention on the TNF axis, it's important to take a look at all 6. So we're going to do a little TNF primer. Okay, so membrane TNF is expressed on the surface of immune cells. It's cleaved off to form soluble TNF. So membrane TNF is the precursor. There are 2 receptors for TNF, TNF-R1 and 2. They're cleaved off to make the soluble forms of the TNF receptor, STNF-R1 and STNF-R2. Soluble TNF is cytotoxic. It's inflammatory and sometimes cytotoxic, can induce apoptosis, but only through the TNF-R1 membrane receptor. That's good in the sense that it is the cornerstone of immunity when it is focally delivered. It's bad because it leads to inflammation when it is non-focal, systemically elevated. STNF cannot signal at all through TNF-R2, so we can discard that. The soluble TNF receptors don't transduce a signal because they're not connected to a cell. They are simply inhibitors of STNF. That's good in that they create immune privilege, particularly fetal immune privilege. Pregnancy depends on STNFRs. It's bad in that it's the defense of all solid tumor cancers. It's an undruggable target. A drug against soluble TNF-R1 would also bind and block membrane TNF-R1. That pathway must be kept open for immunity to function. And it rises with age. These targets rise with age. Membrane TNF is different than soluble TNF. It ligates TNF-R2. That is an anti-inflammatory signal. It promotes survival, cell proliferation, and tissue regeneration. It's good because it's an inflammatory control pathway, and it has some bad elements in that it's involved in gastrointestinal diseases of different types. Notice that these 2 pathways have oppositional effects. STNF ligating TNF-R1 is inflammatory and induces apoptosis. Membrane TNF is really the opposite of that. It's anti-inflammatory. Why is that important? Well, we'll get to that in a minute. I just want to clarify that STNF-R1 and 2 also bind and block membrane TNF. So there are 5 approved drugs against TNF. It's the largest drug class of all. I think I have it here. It binds both STNF and membrane TNF without distinguishing between them, and it generates $42.2 billion a year. Given that the 2 pathways are oppositional, of course, the results of the drugs are chaotic. Sometimes they're helpful, sometimes not. There's one more capability of TNF component. STNF-R1 can also reverse signal through the membrane TNF ligand on the surface of immune cells and induce apoptosis, which is really interesting. We'll get to why that's important. So those are the players. And we're developing nanots against the 3 soluble forms. All right. So for cancer, soluble TNF receptors are intricately involved, we believe, in all solid tumor cancers. We know that STNFR depletion by apheresis is powerful because there's already dramatic clinical data in late-stage human cancer patients. This is apheresis data. Now, nanots are a better way of depleting this target, but it's still great to have a predicate in humans. So there's a clinic in Germany. I've been there a bunch of times. It's called the International Immunology Foundation. They treat patients via apheresis of STNFRs. It's a monotherapy. They're not combining it with checkpoint inhibitors or anything else. It's an all-comers study. They treat any solid tumor cancer patient that comes in the door, and they've generated an ORR across over 100 patients of 60%. With an all-comers study where you're dealing with every kind of cancer, you really can't go above about 20%. 10% is considered extraordinary. And this is, the ORR is the sum CR, complete response, and PR, greater than 50% tumor reduction. So 60% is phenomenal. Okay, I know I'm American. You don't have to run for the exits. It's a different kind of trigger. I'm talking about a very dramatic, explicit tumor image. So if you're squeamish, look away. 3, 2, 1. Okay, this patient had a late-stage metastatic melanoma that was inoperable. The patient had already failed standard of care. He had 24 treatments by immunopheresis that led to a massive necrosis of this tumor. This was not, there was no drugs involved. It was achieved entirely by taking STNFRs out of blood. So we've made Nanots against mouse STNFRs and tested it in a mouse model of triple-negative breast cancer. We saw 5 times the number of metastasis-free survivors in this study versus the control arm, which was saline, and also a mouse equivalent of Keytruda, which is anti-PD-1. So it shows that Nanots can drive dramatic therapeutic response in metastatic triple-negative breast cancer, again, just by taking something out of blood. And we're progressing this to human studies with MGH in Boston under Keith Flaherty, who's kind of legendary in immunotherapy. He co-founded Loxo, which sold for $8 billion. Okay, I want to talk about MS. MS is it's an inflammatory disease, and it's known to be driven by TNF, but the drugs against TNF make it dramatically worse. A clinical trial of that was canceled because it was having a catastrophic outcome. So it's helpful to understand these targets and the role they play in disease and the way Nanots have a distinct role. We have a dual Nanot strategy for treating this incurable neurological disorder. So MS is progressive. It hits 1 million patients a year in the US or 2.8 million worldwide. Poor prognosis, loss of motor and cognitive function, very serious pain, low quality of life. There's a relapsing, remitting phase that lasts 5 to 20 years, followed by a progressive neurodegenerative phase. There's no cure. Existing drugs slow it, but they don't cure it, and there's a very high side effect burden. And it's a huge commercial opportunity because of the unmet need. So here's the pathogenic process, and I'm going to introduce the characters. So in the periphery, there are T cells and macrophages. In the CNS, there are microglial cells. Those are basically macrophages with their potency turned down because of the delicate tissue in the brain. There's astrocytes that maintain neurons. There are ODG cells, oligodendroglial cells. They're responsible for maintaining the myelin sheath on the axons. Myelin sheath is like insulation on a wire. And when you lose your myelin, the wires then short circuit. So there's an initial trigger, and that stimulates, and it's STNF-driven, soluble TNF-driven, that stimulates macrophages and myelin-reactive T cells to differentiate into a subset of Th17, Th1 cells. This causes a huge release of STNF that permeabilizes the blood-brain barrier. Macrophages then infiltrate through those gaps, and further STNF secretion will directly attack the ODG cells through their TNFR1 receptor, will kill them, and that will STNF will also stimulate microglial cells and macrophages. They will attack the myelin sheath, and you get demyelination, which is characteristic of the pathology. There's a mitigating process that runs in parallel to the pathogenic process. This is mediated by mTNF, membrane TNF-bearing immune cells like Tregs. It's suppressive of the cells that are delivering soluble TNF, and it undoes the STNF. And it happens inside the CNS as well, mediated by microglial cells and astrocytes. And it also will help to regenerate the ODG cells, which leads to remyelination. So the big question is, why does the pathogenic process overpower the mitigation process? And the answer is, STNFR1 is in there as well. The same molecule we're depleting to treat cancer is in there blocking membrane TNF, which is suppressive and rejuvenating of these cells. So our strategy is to develop nanots against STNFR1, which we're doing anyway for cancer. I'm going to go through this kind of quickly. And these nanots will deplete STNFR1, deplete soluble TNF as well. 1 nanot against soluble TNF, 1 nanot against soluble TNFR1 to undo the main pathologic elements and rebuild the myelin. They promote the recurrence, the proliferation of the immune cells that rebuild that. So I'm out of time. This is only going to take about 1 minute. We also are targeting age-related soluble targets that disrupt endothelial barrier function. We think this is a core age-related pathology. So there are 3 molecules in particular, CXCL11, CXCL9, CCL11, also eotaxin. These are chemokines, and acid sphingomyelinase, which is an enzyme. And all together, they permeabilize the cell membrane, which leads to inappropriate movement of molecules and cells from tissue to circulation and circulation to tissue. If you think about everywhere that the vasculature goes, which is everywhere in the body, if you dysregulate the vasculature, as these molecules do, you're going to see pathology throughout the body. I'll skip our platform. I mean, it's everything we've talked about and can be combined to treat sepsis and inflammaging as well. John, who's our CTO and IP expert, is here, so say hello to him. He's responsible for our amazing IP estate. And, oh, I saw this in America, and I thought, that's not woke at all, so I ought to put it in my presentation. So that's it. Thank you very much.
Congratulations, look like great data. I'm a neurologist by training with a large MS center in my department. Actually, with B-cell depleting drugs or anti-CD20 drugs, we are very successfully treating relapsing remitting MS. Our problem is actually primary progressive MS. So my question to you is this really running also in primary progressive MS, which would be fantastic because we don't really have to offer much, and CD20 depleting drugs are not really working there. And coming from Germany, I highly invite you to include our center into your clinical studies.
That sounds great. We love that idea. So we'll talk after the event. So we only know what's in the literature, and there's been a lot of new stuff that's come out on MS in the last 2 years, particularly focusing on the differential effects of soluble TNF and membrane TNF, and the need to deplete soluble TNF without disturbing membrane TNF. And one of the theories is that as this ramps up, the actual transition from the relapsing remitting phase to the progressive phase is linked to the inhibition of membrane TNF. So STNFR1 in reverse signaling into mTNF-bearing cells, that seems to correlate with the transition to the most acute neurodegenerative progressive stage. So we'll see. We'll see if that holds up.
How do you get the selectivity to bind specifically the compounds that you want?
Sure. We just build on the science of antibodies. So our current prototypes use antibodies. So antibodies, the antibody art is that they are perfectly selective for their biochemical target. The problem is that the soluble forms and the membrane forms are biochemically identical. So that's where the selectivity of antibodies falls short. It's not a biochemical problem, it's a geometry problem. We solve it with geometry, not with chemistry. Now we are transitioning from antibodies to nanobodies because they're much more compact. We can get a lot more of them on the surface of a nanot, greater avidity. greater efficiency per nanot.
Hi, I'm just curious about senescent cells. Can you use this to select markers on senescent cells? And
I'm glad you brought that up. So senescent cells, it's the same mystery with senescent cells that we have with cancer cells, which is they're grossly abnormal. Why does the immune system put up with that crap? Why doesn't it clear them out? Well, it turns out that senescent cells defend themselves with the same soluble immune inhibitors that cancer uses. You read Judy Campisi's work, you look at what's in the SASP, it's packed full of immune inhibitors, including both STNFR1 and STNFR2. So we think the same technologies we're developing to treat cancer to strip away the tumor-generated immune inhibitors will also strip away senescent cell immune inhibitors and promote a natural clearance. On top of that, senescent cells are cranking out all the sorts of crap that drive inflammatory disease. The inflammatory cytokines are upregulated, and that promotes further senescence, as well as promoting inflammatory disease. So the nanots against the inflammatory cytokines should treat the consequences of senescence, even if we are unable to clear senescent cells.
Thank you, Martin. I have a question about Alzheimer's disease. Can you maybe clean amyloids with this technique?
We have a white paper on that. We have white papers on everything I've talked about. They need NDAs, but if anyone who really wants to read our white papers, we'll do an NDA. We can bring you up to speed on that. But we have a white paper on Alzheimer's disease. We could potentially... there is evidence suggesting that the formation of plaque is driven by the failure of clearance of amyloid monomers from peripheral circulation, and that to promote the clearance of monomers via a nanot strategy would either reduce or potentially even reverse plaque formation. There are also plaque-busting drugs, like EAPs, which break up plaque into monomers. They're not effective clinically because they just reform into plaque. But combining a drug that breaks up plaque with a drug or with a nanot that collects the monomers, that could be curative. On top of that, there's pretty good evidence suggesting that Alzheimer's is fundamentally inflammatory, that it correlates with the emergence of the elevation with age of these inflammatory cytokines. So depleting those on a regular basis could reduce or prevent the emergence. Thank you, everybody.
Thank you very much. It was super inspiring. All right.